All right, welcome to our glycolysis series, part 2.5, which sounds a little weird, but this is a new video that I'm inserting to kind of highlight specifically what you're expected to know about the individual steps. So in the first video, we did an overview of glycolysis. In the second video, we looked at those individual steps. And part of those videos I made this semester, part I made last semester, and I've kind of refined what I would really expect you to know and think about. And so that's the purpose here, is to talk about what you need to know, what you need to understand. So a couple things. This was supposed to be the last slide in the first video, and we did talk about it. So we talked about some terminology, committed steps and regulatory steps. So, committed steps. First irreversible step in a metabolic pathway under physiological conditions, after which the molecules are committed to the pathway. So, if we look at this example, going from some substrate 1 to 2, and man, once it gets here, it could either do this reaction or this reaction. And then it could go on to 5 and 6. Well, if we want to talk about the committed pathway, for the biosynthesis of compound 6, this would be that committed step. Okay, assuming this is a one-way irreversible step, after this point, it doesn't have any other options. Okay, so this would be the committed step. Regulatory steps are enzymes, or regulatory enzymes, or enzymes that respond to cell conditions and tell your processes to either speed up or slow down the reaction. And we will look at those in depth. We're going to talk about it in a moment very briefly, but we'll talk about them in depth in the fourth part of this video. All right, so if we look at our 10 steps of glycolysis, in this kind of more detailed picture, if you come in here and you look closely, we can blow some of these up, you'll see some of these are one-way arrows, versus some are two-way arrows. So glucose 6-phosphate to fructose 6-phosphate energetically can move in both directions, versus glucose to the glucose 6-phosphate with that hexokinase is really a one-directional reaction. So the regulatory enzymes, we're going to look for those steps with those one-way arrows. Those tend to be regulatory. So step 1, hexokinase, would be a regulatory enzyme. Step 3, that phosphofructokinase, and step 10, the pyruvate kinase. All are regulatory. And again, we'll talk about those in depth in the fourth video. Step 3 is the committed step for glycolysis. So products, once we get through this step, the fructose 6-phosphate to the fructose 1,6-bisphosphate, really these things are committed to continuing on down the path to pyruvate. Okay, so then really what I want to talk about in this video is these steps. We talked about these in depth in the second video. And the question I get a lot is, well, what do I need to know? Do I need to memorize all this? Do I need to be able to draw the structures? What do I need to know for these 10 steps? And so as we look at this, let's talk about that. So you should know what happens in each step. So if I come up here and I look at step 1, I remember that I have a phosphate being added to my glucose, right? So I start with glucose, I end with that glucose 6-phosphate. Remember that in that first video, we had in some plain print, it was telling you exactly what happened in each. So you can use that as a review. But you should also be able to look here and say, wow, the only thing that's changed is that phosphate. Okay? Based on that understanding of the starting point and what's happening in each step, if I gave you product molecules, you should be able to say, oh, this one, that would be the end of step 1. Because I know step 1 started with glucose, my 6 carbon sugar, and I know that I added a phosphate. Okay, also based on your understanding of what happens in each step, you should be able to identify the correct enzyme from a list. So remember that enzymes are named based on their function and their substrate. So for example, in step 1, it's hexokinase. All right, well, kinase. Kinase should make sense. We're adding a phosphate, we're transferring a phosphate to the molecule, hexokinase. We've got that 6-carbon sugar. So you should be able to pick this out of a list. I wouldn't, well, this one actually you will need to memorize, but many of them you don't need to memorize the specific name, but you should be able to determine which one is correct. And you should know the important co-substrates. So for example, up here, we're pulling that phosphate from the ATP. So we start with some ATP, we end up with the ADP. Okay. In addition to those things, you should know which steps are the regulatory steps, and we will talk about that again a lot in the 4th video. You should know the specific names of the regulatory enzymes, so hence hexokinase. That's why I would expect you to know that one. You should know which step's the committed step. We just looked at that, and go back and look and see which step has the redox reaction. Now, I'll note that as we go on and continue our discussion, we're going to talk much more in depth about regulation and some other details that you'll also need to know. But looking at what we've learned so far and looking at those individual reactions that we looked at in the last video, these are the types of things I expect you to know about those. All right, one other thing that I wanted to come back to in this first step, so our regulatory enzyme hexokinase, we can see that this is largely one way, right? It's heavily shifted to the right. And I want to talk just a little bit, if you remember when we talked about enzymes, we talked about the idea of induced fit. And this is a great example of that. So this is our hexokinase protein. All right, and when we've kind of got 2 lobes, and in the absence of glucose, so here, glucose will fit into the active site, but when there is no glucose, you can kind of see that there's this opening here. When glucose comes in and bonds, and ATP bonds, they come into the active site, and this molecule, this protein, wraps in around them. Look how this is closed off versus how it was open, right? So glucose and ATP enter the active site, and that changes the conformation, and this thing wraps in and closes off. And that does a couple things, right? It keeps these things in there until they've reacted. It also keeps water out. Remember, this is in a highly water-filled environment. Keeps water out. If water could move in there, it's possible that we could end up taking the phosphate and adding it to water instead. So we're going to protect the reaction we want because of this induced fit model, right? So the enzyme is changing to fit our substrates and help the reaction proceed. All right, that is it for our little extra video here.